This is our poster presentation on the relationship between BDNF and PGC1-α in the human brain, implications for the role of exercise in Parkinson's disease and depression. My name is Heather, my research partner is Ryan, and the research professor that oversaw this was Dr. Stow.
So for our research question, we chose to study the relationship between BDNF and PGC1-α in the human brain and examine its implications for the role that exercise plays in Parkinson's disease and depression. We therefore hypothesized that a relationship exists between BDNF and PGC1-α, and that this proportional relationship can be used to study its impact in individuals with diagnosed Parkinson's disease.
For some background information on Parkinson's disease and the BDNF PGC1-α pathway, Parkinson's disease is known to lead to the degeneration of dopaminergic neurons in certain areas of the brain, such as substantia nigra and hippocampus. This is so detrimental because this loss in dopaminergic neurons has cognitive as well as physical defects, such as eventual loss of motor function seen in people with Parkinson's disease. Furthermore, individuals with diagnosed Parkinson's disease have higher rates of depression as compared to healthy individuals, which further promotes our interest in investigating this pathway. BDNF, also known as brain-derived neurotrophic factors, are involved in neurogenesis and is part of a larger biochemical pathway with PGC1-α. Research has shown that exercise can promote this pathway by increasing the concentration of BDNF PGC1-α in both brain regions. Therefore, in individuals with Parkinson's disease, we see a downregulation of PGC1-α BDNF pathway in both brain regions. However, this pathway's overexpression in the brain can lead to neuroprotection of these dopaminergic cells and reduce the damages seen in patients with diagnosed Parkinson's disease.
So for the methods, to assess the potential correlation between BDNF and PGC1-α, we use the Allen Brain Atlas database to obtain samples from 6 postmortem brains. These samples were taken in the form of z-scores and were extracted from the hippocampus and substantia nigra regions. The Allen Brain Atlas used laser dissection to extract postmortem brain tissue in the form of mRNA. The extracted RNA was then hybridized to cRNA and converted into a microarray chip. The hybridized data on this chip is what the Allen Brain Atlas used to quantify z-scores for each brain region. We therefore collected the z-scores from 2 separate probes in the hippocampus and substantia nigra using all 6 postmortem brains, eventually obtaining 12 samples.
After running a correlation analysis through the SPSS statistics software comparing the 2 probes, we found a significant negative correlation between PGC1-α and BDNF expression when both the substantia nigra and hippocampus were analyzed. However, no correlation is found when the brain regions are analyzed separately. In Figure 3, we created a scatter plot to demonstrate the correlation between BDNF and PGC1-α among one of the 2 probes. Figure 4 is the correlation table that summarizes the same relation between all probe combinations in both brain regions. This significant negative correlation that we found was different from what we originally predicted. Instead of seeing a strong positive correlation among BDNF and PGC1-α, we see a significant negative correlation. This negative correlation, however, has been seen in select studies studying the hippocampus and substantia nigra.
And so to explain this unique relationship between BDNF and PGC1-α, we can look at a couple of reasons. First, we only collected mRNA data, while the data in the research articles was analyzing protein levels. This can help explain the negative correlation we saw if high levels of mRNA and PGC1-α were observed, but low levels of BDNF were seen due to BDNF's downstream location along the biochemical pathway. Compared to the substantia nigra, which had average levels of PGC1-α and BDNF, we saw abnormally higher levels of BDNF in the hippocampus, and also we saw lower levels of PGC1-α within the hippocampus as compared to the substantia nigra. Another potential explanation for the odd correlation seen between BDNF and PGC1-α could be the contribution of oxidative stressors, since previous research has shown that oxidative stress has a significant impact on the biochemical pathways of the brain. Abnormal levels of these oxidative stressors in the hippocampus specifically could have contributed to this negative correlation. To more accurately study the PGC1-α and BDNF pathway, we would have to look at either mRNA or protein levels in a specific brain region, whether that brain region be the hippocampus or substantia nigra.
Thank you so much for listening to our poster presentation. If you have any further questions, please feel free to ask.